Oh, here we go. Did that doofus see his shadow or not?
He did. He saw his shadow. Izzy and I watched it this morning, and we were very upset. I was very upset. But he always sees his shadow anyways.
Most of the time.
I can't even remember the last time he did it.
Oh, yeah.
How long ago?
I thought maybe last year.
Last year?
Maybe. I mean, it was an oddball. Go back and look. Yeah, I could be wrong. We once went there and to see it. And if you haven't been there, it's not a trip you really want to take. So I am not being. I would not recommend Punxsutawney as a destination. We
24.
oh 2 years, 2 years ago. Okay.
2 years ago?
Yeah, 20. Hey,
Was he lying about it? He was.
always. Does this look like spring?
Right?
WTF?
Period. WTF, yes. You are so real.
As opposed to being not real. Sorry,
As
I'm
opposed to being fake.
okay. Do not use any colloquialisms that I am unfamiliar with, which is all of them.
Okay, are you familiar with that one now?
Yes, I'm getting better.
Okay, good.
You have to write them down and add to me the context that I've used.
We will do that.
I appreciate that.
Of course.
All right, let's get ready to start. Nice talking to anybody. Here we go. It starts to get hard now because the way to frame it is this is a cell with which you are not familiar. Okay, you haven't seen this cell. I mean, you may have seen this cell before, but the reality is for the vast majority of people, we're so used to seeing our cell, the cell membrane, the nucleus, and all that stuff, and the chromosomes and blah, blah, blah. Not the same. And that's really, to me, this the appropriate frame to look at this. I mean, their structures it's going to look complicated, but it's a bad illustration. It's comparatively simple. You hear me describe it frequently as elegantly simple. And basically, it's dedicated to reproduction. That's all it does. They reproduce. The stuff they give off are generally toxic to us if it's a bad bacteria, helpful to us if it's a good bacteria. The vast majority, again, are your friends. And other than that, all they do is make more bacteria and very quickly. I mean, when you're doubling you're going to see every 20 minutes to every half an hour, you get tremendous numbers in very, very brief amounts of time. Moving on. We'll answer all those. So we look at it from the outside in. What we'll probably at least touch on today or identify today as a transition is aptly referred to as the cellular envelope. And that's when we're going to start to look at the cell wall and the related structures. So we're going to look, the cytoplasmic membrane, thankfully, is basically like ours, a phospholipid dilator. The cytoplasm is cytoplasm. It's not identical to human cell cytoplasm, but it's certainly similar in a lot of ways. And ribosomes, again, other than that density difference, the 70S versus 80S, and the way they're constructed are ribosomes, except there's no such thing as a membrane-bound ribosome because there's no membrane inside. And the cytoskeleton is much more limited, as you will see, than we have. We have that elaborate one with intermediate filaments and microfilaments and microtubules abounding. And they only have some tiny microfilament-like things. And then the idea, which is a very significant difference, the chromosome. Notice the singular tense. But by and large, there's one bacterial chromosome, and it's distinctly different configurationally, if that's a word, in the way it is arranged than ours, even though the DNA is DNA. And RNA is RNA. So that's where the similarities are, other than the density and the construction of the RNA. And we'll look at the cell wall, which we certainly don't have, and we'll look at the glycocalyx, which is remarkably different in a lot of ways. And then the way that they interact with the environment and the way that they get around involves a lot of what are called external appendages. This is a significant difference. I made a big deal of it when we had A&P, and the vast majority of you had me for A&P. Okay, is effectively, there's no such thing as flagella are really inside the cell. They look like they're protruding, but they're surrounded by membrane. Cilia. They look like they're protruding, but they're surrounded by membrane. We call them internal cellular appendages because they're sheathed in membrane. They're really inside the cell. These are more like hairs. And we have ones that are long, flagella, ones that are shorter, pili, and very important in some specifics of certain kinds of pili, and fimbriae, which are like little bristles that attach. So even though they're very, very similar, they're constructed a little bit differently, as you will see. Okay, they'd have an outer membrane. I'm not worried about nanowires and nanotubes. Plasmids, I touched on ever so slightly. We talked about just a little introductory remarks about how we do genetic engineering. Plasmids are part of the key. Inclusions, really more for identification. And so I'm going over these laundry list style, and then we'll look at them more deeply as we go through the PowerPoint slides and the discussion thereof. So inclusions are things, some cells have different things in them, and you may start to notice some of these in lab, depending on, and I suspect you'll look at your plates and maybe you'll start to you probably do basic staining, I would think, this week. So you start to stain things, and you might see, things that look like they're inside these tiny bacteria. Endospores, extraordinarily important. I'm not worried about the micro compartments, and we're not doing archaea other than what I told you about them previously. This is kind of what E. coli would look like. E. coli is a rod-shaped bacteria. It generally has one long flagella. You're going to see the more flagellae, being plural, that it has, the more it can move around. And E. coli can move. And basically, it looks like it's more complex than it is, but the complexity is all of these layers. And we'll look at the external layers. We'll look at the cell membrane. We'll look at the peptidoglycan. And I can't, maybe if your eyes are better than you all must be. I've got to make an eye appointment in a couple of years. I can't even read all this stuff, S layers, but if you're feeling ambitious, you can read it, but we're going to go over it again anyway. But really, these are basically the ingredients here. If you look in the cytoplasm, not all cells have these kind of, whether they have granules or little nuggets of stuff like that, but they all have the chromosome and they all have ribosomes. That's at a minimum, and they're all inside the cytoplasm. After that, it doesn't have to be a whole lot else. They're sort of giving you everything here. So this is where effectively I start as we go over each of these components. They're on the handout I have that I posted. A lot of that is on there. I took that from the other textbook that I use, from the other PowerPoint sequence. And I did that outline. It hasn't changed for sure, more than 20 years ago. So bacteria function as independent, single-celled, unicellular organisms. So organisms is correct. Sometimes they are completely independent, and a lot of them functions as colonies. That's true. Biofilm. I'm curious, did any of you, when you were doing those samples, did anybody do like inside your mouth or your teeth? Anything like that? Okay. On your teeth is dental tartar. We all know that. We go to dentists, flossing, and stuff like that. It's all about the tartar. That's a biofilm. If you get around and you stain that, when you probably will, what you will see is a vast amount of organisms. Our mouths are a cesspool of organisms. And it's not because you didn't brush your teeth or you didn't do this or it wasn't about mouth care. It's worse with bad mouth care. Just saying that. But understand that it's extraordinarily filled with them, and it's a great way to look at them. Biofilms, what dental tartar is basically, as you will see, it uses this outside material that protrudes or extends from the cell wall. And really all of it is a series of different kinds of sugars. So you're going to see the term glycocalyx, which means sugar shell. All our cells have, to some degree, glycocalyx in us. But when we put them here in bacteria, a lot of them form this sort of sticky kind of material that adheres, and it forms colonies, and it really almost is almost an environment of its own. That's where dental tartar is pretty interesting when you look at that. And yes, even though I'm not worried about naming things nanotubes, a lot of the bacteria is set up it's almost a community of bacteria. Sure, of course, dental's harder. Not, and it's not something I'm going to be asking you about, other than the fact that it's a biofilm. Okay? The outside is aerobic. We inhale and exhale, so the bacteria have to be able to thrive in air. Against your teeth, the air doesn't get through it. It's anaerobic. And the bacteria that causes cavities is a bacteria that doesn't like air. It's actually a variant of Streptococcus. Streptococcus doesn't do well when it's exposed to the air, and that's the, quote, karyogenic is the fancy word for cavities. Makes cavities. That's why getting rid of the tartar as much as you can limits the likelihood of cavities for those who, everybody's, what I understand about dentistry, you could fit into a tiny thimble with a lot of room left. It's short for nothing. When I do AP2 and we get to digestive, when we get to the teeth part, I go, I don't do teeth. I just skip it. I don't want anything to do with the teeth. So bacteria are tiny. Viruses are much tinier. So when they talk about a micron, ballpark like a red blood cell, probably about maybe 6, 7, 8 microns in diameter. Okay, give you an idea. So they're smaller than like a red blood cell. So they come really in 3 major forms. Cocci, which are little spheres. Okay, you don't have to worry about the dimensions. Rods, which could be longer or could be wider, and rods are very interesting because you get long, skinny rods and classically shaped rods and rods that almost have sort of a mixture of a rod and a sphere. We actually call it coccobacilli. The fancy name for rod, I wrote it up before, with a small b, is bacillus or bacilli for the plural. So when we use a small b, ACILLUS, or if there's more than one, we take out the US and put an I. That means a rod-shaped bacteria. If we use the big B, bacillus, that's a genus. Remember, the endospore is the one you're going to use in the lab, the Bacillus subtilis. Anthrax, Bacillus anthracis, bacillus. Big B versus small b, so letters count. Okay. And that can confuse people on exams. Okay, that's why I'm pointing out to you. Every little thing, not telling you something that I hadn't talked to you about before. Most of you could be a lot better test takers. And a lot of it has to do with question reading. I mean, we're going to work on that test taking anxiety, which is rampant for whatever reason. We'll work on it. Just so I'm trying to give you this heads up and make life a little bit more easy for you in this semester. You'll have enough going on with other stuff. Okay, so bacteria have that size. Pleomorphs involve different kinds of variations that are there. We'll get to them. Pleo means many, morph means shape. So when you hear about pleomorph, it can be different shapes. And a lot of that have to, and there's a specific one that we will come to. So the coccus, okay, spherical or ball, they're not, as you can see, some of them are beautiful circles. Some look like little coffee beans. And that's one of the ways we identify them. That's more about lab stuff. So you're looking at the one that's the easiest one to see, and you see it all the time. It's called Staphylococcus. What coccus means, what I beg your pardon, what staphylo means is a bundle. Okay, and it has to do with how they replicate. So if you take coccus and then it replicates linearly, and here's another one, and another one, and another one, they tend to form a chain. The name for that, very famous, strepto. So when you get a strep throat, people have a oh, strep throat has got stripes in it or something like that, a certain way. No, it's all about it has to, you're describing the bacteria. It's named for its shape. Okay? So strep throat, strepto means chains of. Sometimes the chains don't assemble and you get little pairs. Okay? They're called diplo. The bacteria just, and we're going to do this a lot when we get to respiratory. a very famous strep throat bacteria. And it's not only throat, it's also skin and other areas. Streptococcus pyogenes, chains of cocci that produce pus. Fancy sounding name, very straightforward description. Okay. Staphylo means clusters. So when you have Staphylococcus epidermidis, we all know what the epidermis is. These clusters of cocci that we find on the skin. It's on all of your skin all the time. 100% of your skin, 100% of the time. So once you know those little root words, it's easy. As long as you remember a little bit, and we'll get into what they're like. So the bacillus, as you can see, not the genus, are varied. So you can have ones that are club-shaped, and that's all in the lab, how you identify them. You're not going to go... you'll undoubtedly do unknowns. The unknowns will be straightforward because you know what to pick from. You'll have a list probably of all the unknowns because all the bacteria you study. But these are all little clues that in a bacteriology lab, in a hospital, where they look at the shape. What do they look like? I mean, traditional rods like that, pretty much E. coli. This club-shaped kind of a rod, interesting. There could be an endospore hiding in there. Skinny ones, I think Pseudomonas. We'll see even chains of rods. That Bacillus subtilis is a kind of streptobacillus because it has chains. This is kind of what I do when I do the lab. I did it yesterday. So here's a photo of Legionella pneumophilia, a rod. You can see there's some degree of adherence between them. Legionella pneumophilia is very famous. It led to something called Legionnaire's disease. Legionnaire's disease, very interesting disease. And again, you'll see, like I keep saying, we'll look at it in respiratory. It gives you an idea that respiratory is a really juicy chapter filled with a lot of goodies. Thankfully, we don't get into the second half, of course. So Legionnaire's disease. And that's right, I was there when it was first came to our attention in Philadelphia. Who was that guy who was always there when the things happened? He'll come to me. Tom Hanks won an Academy Award for it. He'll come. Moving on. So here we have some interesting ones. Sometimes they're curved. Vibrio. Vibrio means curved or comma shaped. So they're not necessarily straight. The most famous Vibrio is Vibrio cholera. It causes a dreaded disease that you see a lot with contaminated water supplies, and particularly, and again, after natural disasters, war-torn country, called Vibrio cholera. So Vibrio means comma shaped. And then you get all these sort of spiral forms. They're curled, and they can be very curled, or they can be only slightly curled. Okay, so spirillum is sort of the spiral forms, general sort of way we refer to them. So they can be, and the one that we really, again, I alluded to it last time, look like a corkscrew, spiral keys. There are two really famous ones. One causes Lyme disease. We will have to know about that down the road. That is called Borrelia. I'll have to learn it now. Fun name. Burgdorferi, because the guy named it after himself. Okay, and the other, I was just reading an article about syphilis. Beginning around thousands of years. It's not clear how we found it. I mean, we thought that syphilis might have come from the New World and brought to the Old World, but I think it's more likely the other way around. So syphilis, SYPH, is caused by a very famous thing. It's a capital T. Treponema pallidum, which actually means the pale thread. for fun. We're going to get to all of those, but now here, this is one. Jejunum is the second half of your small intestine, so it's found in the jejunum. It's called Campylobacter. I don't know. Bacter is pretty obvious where that comes from. Interestingly, that can cause food poisoning. So some gut bacteria that sometimes fecal contamination, whether it's from us or from some other animal source, into the food supply. So that's one that we sometimes see, and we love to nickname these things. They call it Campylobacter jejuni. And so here's the spirochetes. Bless you. And you can see the difference. Here's a micrograph blown up 7,500 times, and you can really see the corkscrew appearance that's there. It's quite distinctive. And it's interesting the way they move around. And then you have the oddballs. Streptomyces, it's in the soil, and it actually looks kind of like what mold looks like, almost like bread mold. And what happens is the replication comes at these ends, and they're actually it's almost like a spore forming bacteria, but it's really not there. And it's very, very famous because a ton of antibiotics come from this. When you get the antibiotics, you'll see that it says streptomyces. Side note, I've been going in the nursing building and going downstairs on the first floor, and I saw one of the whiteboards. They must be having a farm exam about antibiotics. It's a bunch of stuff that I teach. I was like, I got to tell you guys about it. So I'm like, I taught them that. Pay attention. This could be helpful down the road. Okay, don't pay attention. Be like my kids and don't listen to me. So you have diplococci in pairs, groups of 4, staphylo clusters. Okay, streptococci change. I'm not worried about the packets, Sarcina. We don't work with them. And it's all about how they replicate. So a lot of times, streptococcus can also be just pairs. So streptococcus pneumonia has 3 different names. That's the leading cause of bacterial pneumonia in adults. It's also called Diplococcus pneumonia, and we even gave it its own name, calling it pneumococcus, generically, just one thing. So when you hear about somebody, I had a student email me who had gotten pneumonia, more likely viral pneumonia than bacterial. But when you think about bacterial pneumonia, the first one I think about is Streptococcus pneumonia. Here are the little packets and the groups, but the big one is Staphylo here. Staphylococci. This is unique. Interesting. Unique is probably the wrong term. These are bacilli, okay, with ends attached. And I've seen, like I said, the streptobacilli, and sometimes you see them in pairs, are Bacillus subtilis. This is called palisades. Okay, cells of a chain remain partially attached. It's like when they don't completely separate, and it forms these interesting patterns. Back in the day, now they call them palisades. They used to call them Chinese letter patterns because it looked like the Chinese calligraphy characters that you would see if you go, at an Asian restaurant or something like that, and you're able to, see the things next to whatever you're going to order. You're right next to that General Tso's chicken. There'll be calligraphy, and I don't know anything about it, but it's and now the species is called Corynebacterium. By the way, that species is on your skin all the time. That species is in your respiratory tract all the time. And oh, by the way, you're all immunized for a very famous Corynebacteria that they're all kind of judged against, called diphtheria. The ones that are not diphtheria, we have a generic name that you'll learn about called diphtheroids. That OIDS suffix means looks like or kind of, but not the same. So yeah. I don't care. Motility and mobility, important stuff. Good child. Okay. Flagella and axial filaments. Okay. They get around by movement. And so normally they're flagella more than anything else that do the movement. They don't all have them. And believe me, they don't all move. Some of them, like Staphylococcus, very, very common. Streptococcus, very, very common. Really aren't what we call and in the lab, you test for them being mobile, or what we call motile. The motility is the word that we used. So some just stay where they are. If their movement is by other things, they might move in the air, they might move in the water, they might move as we walk around and move them around just by a force, but not by themselves. So different things. The flagella is the primary way, and we use this a lot to determine whether they move a little or move a lot, and also to classify them. And so typically, and this is not like our flagella. You remember in a eukaryotic flagella, in membrane, and what it had at the base was that very famous microtubes, all made of microtubes. There's 1, 2, 3, 4, 5, 6, 7, 8, 9. That 9 plus 2 microtubes. And it's the same for carrier, flagella, and cilia. The flagella in bacteria, totally different. One, they're inside, they're outside the cell, they protrude. And so in prokarya, the flagella are composed of just a chain. They name the protein for the appendage called flagella. It's just protein. Nothing really special about the pattern. For pili, they have a slightly different protein. They call it pilin, P-I-L-I-N. And I'm not even sure if they've even concocted a name for Fimbriae. I call it Fimbriae. And again, we're not doing the nanotubes and the nanowire whatsoever. So it's all about motility. And I'm not worried. And really, if you want to go classic, they have a thing called a basal body, a hook, a filament. Don't care. I just want you to know what they're made of, and they're not the same. They just stick out, pieces of protein that protrude. So we look at, I mean, you can see this is a micrograph, and they're showing you really this combination of the basal body and is really how it's implanted in the membrane and the cell wall, and then the hook kind of angles it away. And our flagella and our cilia, if you remember from me talking to you about it, basically kind of did this. They swung back and forth. There was a whip-like motion. Okay? These spin. And they can spin in either direction. So like almost like a helicopter. Shh, or shh. So we look at the arrangement. Is there one single one? So they look like hairs. The term is trichus. So you have monotrichus. Okay? Lopho means a bunch. Amphi means at the either side. Okay, and sometimes it could be a single one, and sometimes it could be a bunch. And then lastly, peri for all the way around. The reason I'm telling you about these is it's the way you categorize them in lab. The ones that move the most are peritrichus. You're going to see one that you're going to look at in labs. Got a great name. It's a gut bacteria called Proteus vulgaris. And it can cause urinary tract infections. Proteus is peritracheal. It moves all over the place. We call it swarming. So we use the reason I'm sharing this all with you is it does a lot of this for identification. So it helps you in the lab, but this is, frankly, the bacteria all look alike. You may have some different colors. Other than that, they're very similar looking. So we have to look for things that help us to classify them and identify them. Once we identify them, then you can treat them. So, for instance, you can see whether it's a bunch, whether it's single, etc. But remember those kinds of things, and you can just look up the different ones that are there. They're very simple creatures. They're either attractive or repelled. If you're attracted to something, you go to it. We call that positive taxis, T-A-X-I-S. If you're repelled by something, you move away from it. That's negative. So positive chemotaxis, and really, and most of the time it's chemicals. The vast majority. Something in your environment. Hey, you go by something that smells bad. You're moving away from it. I'm over at Oakmont Bakery. I'm going in. the only reason I like to visit my daughter and my granddaughter. They live across the bridge. They're over in the Harmerville area. You go over that bridge, baby, you're at the bakery. But I digress. Okay. You don't like the bakery? Have you ever been there? Do you like it? Yeah, they're my kind of people. Where do you live?
I live in Manhattan.
Oh, you're close to us in Cranberry. Okay. There's no Oakmont Bakery there. One of the many dying malls. That's about the size of it. Okay. So here we go. So positive and negative, and they have limited energy. You and I, if we're going to go get some, okay, I got to get a paper towel. We'll walk over here and get a paper towel. Oops, I got it. I'll walk back, straight line. They don't. Bacteria have enough energy until they have to replenish it. They do what's called run, tumble, run. They go a little bit, stop, and eventually they get there, but it's not a straight line type of action. Very distinctive. And a lot of times they're moving, but they're moving sort of aimlessly. It's almost like in a giant circle. So you'll see this. It has to do with the rotation of the flagella, which I don't get into. So they go straight, and then they tumble, and then they go, and they may go in a different direction depending on the tumble, but eventually they'll get where they're going. And they show it to you. This is no attractant or repellent. The fancy name for structures that move and continuously, but really almost aimlessly. Again, it's more of a lab thing. It's called Brownian. I don't know who it's named for. Brownian motion. But with bacteria, it's run, tumble, run. And that's sort of, and it's either, and if it was repelled, it would do the same thing. It just don't understand their resources are limited compared to ours. They don't generate large amounts of ATP like we do, that we can do a sustained activity for a significant period of time. So, and the other ones that are interesting when you talk about guys like this, these sort of corkscrew shaped, it's, you could do this kind of to move, sort of like, you can take like a string and feed it through something and it's almost, I don't know. We did that in the old days in surgery. It's awful to talk about this. We had to do an amputation. We would use, it was called a giggly saw. And it was sawtooth on like a metal thing. And you sort of, that's kind of the way these guys move. Okay. What happens is they're in what and you'll learn about it shortly. It's called tomorrow, undoubtedly, called the periplasm. And they go along the long axis, so we call them axial filaments. So the flagellae are inside, and because of the spiral shape, it sort of moves this way and that way. And again, that's a distinctive motion. So it's very interesting, and it's typically with the spirochetes, and maybe a little bit with any of those forms. Fimbriae are like little bristles. They're all about adhering things. This would be an example of E. coli adhering to areas in the gut where it's not supposed to be. Okay. And the first step for a bacteria to replicate, it has to adhere. It needs a foundation or a base. So the fimbriae are like, not now, but if you go walking in the fall and you get those little bristly things in your sweater, you kind of go up and you got to pick them off. That's kind of what a fimbriae is. It's almost like almost like a Velcro kind of a thing. Whatever works for me. You're all looking at me. What is he talking about? I always use that analogy. Now, pili, made of pilent, or why do they name it? Conjugation. You can see the illustration. When doctors and scientists start looking under a microscope, the concept, first of all, bacteria, by definition, do not replicate sexually. They're all asexual. They don't do mitosis or meiosis because they don't have the right kind of DNA. They do what's called binary fission. But physicians weren't that, and scientists weren't up on all of that stuff, our understanding today. So what they did, they looked at this structure, this pilus or pilus, being the singular, and protruding, much more frequently seen in gram-negative, which we're going to at least introduce a little bit today in gram-positive. Effectively, and they thought this was like a sexual appendage. You had male bacteria and female bacteria. Not so the case. It's used to transfer genetic material. You remember me telling you about how one bacteria could touch another and another and another, and basically change them without having to have a new generation. So they can transfer genetic material. They look like a fimbriae. They can attach to things. They can assist in attachment. They can act like a flagella. They're very unique. And I don't want to really go into the cytoplasmic things with the oxygen, and it's a little bit different. For our purposes, I want to try to keep it more clinically relevant than anything else. So this is what they look like. So one of these pili, or this pili, they call the F for fertility pilus. And sometimes we call it the sex pilus if the bacteria had one. They said it would, now we say it's F positive. So they called it male and female, but it wasn't the case because it was effectively, it was not a form of sexual reproduction. And we look at it, and we'll see an animation about it. I'll point out where that distinction comes from. You're just changing something. So there's a really good animation. S layer is it's like a chain link fence. And this is neat. Imagine in a hostile environment, bacteria that don't like cold can form something that would keep them intact so they can adapt like you and I can. Bacteria can't go, well, it's cold out. I'm going to get a nice coat. I got to get that sweater. I got to get my hoodie that I'm not wearing today. You're all supposed to be smiling about this. Go ahead. You were making fun of me. I know. Don't look at me like that. I saw that. Probably took a picture. Say, look at this idiot. So, can you do that? No, we can't magically change our exterior. And here's the shell, sugar shell. Easy for me to say. Typically, polysaccharides more than anything else, sometimes glycoproteins. If they're loose and disorganized, we call it a slime layer. If they are thick, organized, and typically large amounts of polysaccharides that almost turn into a waxy, they're called a capsule. Capsules are very significant for disease because how your body addresses disease, we gobble things up. We have macrophages. We have white blood cells. The way that they address invaders is they swallow them and destroy them. That whole thing with endosomes and phagosomes and lysosomes, all that stuff we talked about, that's the way our body defends itself. We gobble our invaders up. Love that. It's poetic. If we don't like it, we eat it. You know, I say I want to be reincarnated as a macrophage, so they have a relatively short lifespan. Big eater. Okay. So the capsule is difficult to swallow. Tuberculosis has such a big capsule, antibiotics can't penetrate it. It is swallowed. They attempt to swallow it by white blood cells. They can't dissolve it. So it goes and replicates. It actually sets up shop in a white blood cell in your lungs. How's that? And not one antibiotic, but at least two. And typically you have to alternate the antibiotics, change them up every 2 months for a year. That's how hard some of these things are to treat. So the capsule, very significant. So here's their diagram, an S layer. Here's the glycocalyx. Here are encapsulated bacteria. You will see these in lab. No problem. And that's a test we do. It's a fancy thing. It's called a negative stain. And just very, very distinctive. Yeast that you typically will work with in lab, making just yeast, brewing yeast, are encapsulated. And they stain very similar like that, except they're more circular than longitudinally oriented. So that's another thing we look for, are capsules. That aforementioned Streptococcus pneumonia, what makes it bad is its capsule. Klebsiella pneumonia, maybe one of the harshest forms that you can see in a hospital, capsule. You get an idea? Capsules in pathogenic or disease-causing bacteria are bad. Disease-causing ability, protects against white blood cells and phagocytosis. Biofilms, plaque can become dislodged, things like that. And this is one of the things, catheters. Still the number one cause of what are called nosocomial infections. That means hospital acquired. The bane of your existence, everybody here, if you're going into nursing, the biggest unit you're going to do right away is dealing with catheters. Indwelling urinary catheters. They are we've improved greatly. We have reduced the infection rate probably 25, almost approaching 50% of what it was when I first started teaching us. So we've learned about this stuff. They used to do a lot more, if you didn't know, they used to do a lot more straight cathing than indwelling Foleys than they used to. Those are not fun to have inside you, by the way. Not fun. Unfortunately, between my stomach surgeries and my bladder cancer, let's not talk about it. So persistent IUDs, pacemakers, other devices. So what happens, orthopedic devices form a biofilm on them that's filled with Staphylococcus epidermidis. Unfortunately, the type it gets filled with typically causes infections inside. So when you hear about artificial joints, you hear about hardware, I've taken it out, it's got a film on it. In my own patients, I've had to pull pins and hardware out of it. Okay, and so they're showing you a bit of how this, and it's pretty good. Here's a surface, this would be a biofilm. The cells start to adhere to it because of that glycocalyx. They continue to divide. Then, along with that, forms almost, you can see almost a colony developing. And that's kind of how it morphs. It has a life of its own. Little passageways in it. Neat. The slime layer is not an appendage. Okay, the hard part. The term I like is the cell envelope. It includes the cell wall. It includes the cytoplasmic membrane. This is the introduction where I just want to give you an idea of the complexity of this topic. And finally, in some bacteria, which you're going to learn are called gram-negative, they have an outer membrane. It's a barrier. The biggest single distinction that you are going to have to be acquainted with when you're dealing with bacteria and you're learning about antibiotics and you're studying pharmacology and you're studying the pathogenesis of disease, pathophysiology, is the distinction between gram-positive and gram-negative. It's the single most important test that you'll do in that laboratory. Okay? They'll go into it in every textbook. It's not that difficult to do. It's difficult to get it right. So these are protective in nature. And well, I like the term envelope. If you can break apart one, you can basically damage the whole. That's what's meant by an envelope. But think of it as a unit. Penicillin works on the cell wall. Once you expose the cytoplasmic membrane, there's nothing to hold that bacteria in its shape. If you can destroy the membrane, okay, it'll kill the bacteria. Trouble is, its membrane is like our membrane. And the outer membrane is a really interesting one. And this is what they look like. Very complex. This is gram-positive. Now, I do not have nearly enough time to go into this, and I anticipated this. Gram-positive have a giant thick wall, 30 to 40 layers thick, of this substance we hear about called peptidoglycan. It sits literally on top of your standard phospholipid bilayer biological membrane. It's held in place. We're going to review all this. I don't go quickly. Called teichoic acids. It's a chunk sitting on there. Okay. How does stuff get through it? It's porous. It's really important to remember. So imagine, as large and significant structurally as this is, if you can stop it from being made with an antibiotic like penicillin or penicillins, think of how successful it would be. Now we get over to the flip side. You wouldn't want to meet one of these on the street. Ugly looking. You have an internal membrane, phospholipid bilayer. You have a thin layer, 1 or 2 layers of peptidoglycan, and then you have this interesting outer membrane that occurs nowhere else in nature, particularly this outer leaflet. It's totally foreign. So much so that if it ever gets where it's not supposed to be in us or any other animal for example, it is a toxin. It's called the LPS or lipopolysaccharide layer. So be here on Wednesday because we'll go over all this stuff and it's a really important meeting. Thank you very much. Get done a little bit early. Don't want to start that without. Aren't you glad you came? Let me have the attendance sheet. Make sure you signed it if you were a later ride.